Haemoflagellates. Under Haemoflagellate, two genus are medically important. That are Leishmania genus Leishmania, genus Trypanosoma. So these organisms have developmental stage in blood-sucking arthropods. So these are the intermediate host. That means blood-sucking arthropods are the intermediate host, and in humans are the definitive host, and they may have a non-human mammalian reservoir host also. So Leishmania species are transmitted by sandfly. The common name is sandfly. Scientific name, there are two under sandfly. There are two flies are responsible to transmit the Leishmania diseases. One is Phlebotomus species, other one is the Leishmania. So next one, the Trypanosoma are transmitted by either tsetse fly, we call the Glossina. It is transmit the African Trypanosomiasis. Other vector is the triatomine bug. These are transmit the American Trypanosomiasis or Chagas disease. The flagellates may occur in a variety of stages in the human host and the insect vector. So when we consider the Leishmania species or the Trypanosomia species, there are so many stages we can observe in their life cycle. So some stages are present in human host, some are present in the vector. General characteristic of Haemoflagellates, they produce by simple longitudinal binary fission. So the reproduction or the propagation is simple longitudinal binary fission. Infective stage always vegetative form. In Leishmania species, infective form is promisticate form. In Trypanosoma species, the infective form is metacyclic trypanosticate form. Transmission occur through biological insect vector as intermediate host. So here, how the human definitive host, human is the definitive host, how they get transmission? They are getting transmission through the biting of the, especially biting of the insect vector. Why we are calling biological insect? Because this vector, in this vector, there are some development of the parasites. That's why we call biological vector. Again, vector we can divide into two parts: mechanical vector and the biological vector. In the case of mechanical vector, they just transmit from one place to another place. The parasitical stage is transmitted by this vector from one place to another place. There is no any development inside the vector. But in the case of biological vector, not like the mechanical vector, in biological vectors, there are some development of the parasitical stages inside the vector. So here, in the case of Leishmania species and the Trypanosoma species, when we consider the vector, inside the vector, there are some development of the parasite. So they are biological vectors. The species are morphologically indistinguishable, but they can be differentiated on the basis of, when we consider the species, so species are normally we cannot differentiate. Morphologically they are similar, but we can differentiate by using some factors, that is the clinical feature primarily. So When we consider the species, each and every species, they are produced the clinical features different. Second one is their geographical distribution. So when we consider the distribution, the area wise they may vary. For example, the some species, for example, Trypanosoma brucei, it is present in the Africa. Then the American Trypanosomiasis we can see in the America. The serological testing, sometimes we can use specific the antigen or antibody. By using that we can differentiate the species. Then culture characteristic also we can use and we can identify it. Then vector. So by using the vector we can differentiate. For example, in the case of Trypanosoma brucei, it is transmitted by tsetse fly. In the case of Trypanosoma cruzi, transmitted by the bug, triatomine bug. But in the case of Leishmania species, they are normally transmitted by sandfly, the phlebotomous species or Leishmania. Then again the reservoir host and other thing biochemical test also we can use to identify is other one immunological tests. By using these characteristics we can differentiate the species, but morphologically somewhat differ to differentiate species. So the different development forms are differentiated in the basis of, so we already discussed there are some different forms are present when we consider their life cycle. So these different forms are differentiated by some special characteristic. One is the presence or absence of free flagellum, whether these stages contain flagellum or not. Second one, presence or absence of undulating membrane. So then we can differentiate by using the undulating membrane whether it is contained this stage undulating membrane or not. Other one, the position of the kinetoplast relative to the nucleus. So according to the based on the nucleus we can use the position of the kinetoplast. By using that we can differentiate again the stages. They have the following main body parts. So when we consider the stages, we can differentiate the some different parts in their body. One is flagellum, other one is the kinetoplast. So these kinetoplasts again divide into two things, pleuroblast and the parabasal body. Other one is the exonum or exostyle, nucleus and undulating membrane. These are the body parts we can see in these stages. Then based on their development in the insect vector and their mode of transmission, trypanosomes are grouped into two. So here when we consider the Trypanosoma species, when we consider the vectors and other mode of transmission, we can divide into two. So Trypanosoma species divide into two things by one is the vector, other one is the mode of transmission. First one, salivarian groups. The parasite develop in the mid and foreguts of their vector and transmitted to the human by inoculation of the parasite. So here in this first group, these vectors are transmit to the human. The parasites are transmitted by this vector through the that means saliva, because the parasite are normally they are developing mid and foregut. When we consider the digestive system of the vector, we can divide into three parts: mid, fore, and the hind guts. So this first group, if the parasite is developed inside the vector, here the mid and fore guts are the main part where we can see the vectors. So here how it transmit: when the vector, they take the blood meals by biting, then the parasite stays transmitted to the host or human. But Stachoralian groups, these parasites develop in the hind gut of their vector. So uh, uh, here the parasites are developed third part, end of the guts. So here the transmit to the man by the contamination bite area with the feces of their vector. So here end part of the digestive system. So normally here if the parasite is present, they may pass with the stool sample which are shaded by the vector. So that time they may transmit when they taking the blood meal. So they are they are making injuries when they take blood meal or whole, right? So that time they take blood. So in same time they shade at the feces. So feces contain the the infective stage. So if it is inoculated, then it is meet the whole or wounded area, it is transmitted to the human. Then the main developmental forms, there are 5 stages we can see in. One is amesticode form, second one promesticode form, third epimesticode form, fourth trypanosticode, fourth, fifth metacyclic trypanosticode form. These are the 5 stages we can see in Haemoflagellates, right? But according to the species, some stages may miss, right? Now we will consider the amesticode form. Normally this is spherical or oval shape. Here we cannot see the flagellum. So here again we cannot see the undulating membrane. And these are the only, when we consider there are 5 stages, this only the intracellular form. Okay, intracellular form mean we can see inside the cell. Without cell they cannot survive. So these stage, that means amesticode form seen in the all Leishmania species and the Trypanosoma cruzi species, but we cannot see in the Trypanosoma brucei. So here when we consider the morphological form, this is oval shape and the cytoplasm color is blue color and a nucleus actually at center. It is purple color, dark red color, and the parabasal body, pleuroblast, axoneme, these all are red in color. Right. Next we will move to promesticode form. This is elongated. Here we can see the single flagellum and a single nucleus at the center. Here also we cannot see the undulating membrane, and the kinetoplast is anterior position, and this form is found in invertebrate host, that means insect, and the culture media. So all of Leishmania species we can see the promesticode form, but in man as a transitional form for the Trypanosoma cruzi. Again amesticode form and promesticode form we can see in all Leishmania species and the Trypanosoma cruzi. And other thing, uh, especially in the case of Leishmania species, we can see the promestigote form in vector, but amestigote form in human. In the case of Leishmaniasis, there are two stages. One is amestigote form, other one is the promestigote form. Amestigote form we can see in human in Leishmania species or Leishmania species, but in the case of promestigote form in Leishmania species, also the promestigote form we can see in the vector. Again, promestigote form and the amestigote form in the Trypanosoma cruzi we can see in the man. Epimestigote form, this also elongated body and it's contain the single flagellum and single nucleus at the center and undulating membrane we can see it is arrives from the kinetoplast, right, and the kinetoplast also just anterior to the nucleus, that means the kinetoplast very near to the nucleus, that means at middle part we can see the kinetoplast. From that the undulating membrane arising and it is freely available in anterior part, that's we call the flagellum. It is found in invertebrate host and in culture media. So, Trypanosoma species where we can see the epimestigote form, one is culture, other one is the invertebrate host, right. So, you have to remember. So, in human we cannot see the epimestigote form. So, only we can see in the host. Then trypamestigote form, this is pleomorphic, that means the morphologically we can see in different sizes. It can be seen as U shape, C shape or S shape. Again, this is also single flagellum, only one flagellum is contained. The nucleus is again at center, single one, right, pink color or purple color nucleus at center, the cytoplasm color blue color, other all parts kinetoplast, undulating membrane, flagellums are in color, red or pink or purple color. So, undulating membrane we can see this undulating membrane arises at the kinetoplast. Here the kinetoplast is at the posterior end, right, behind the nucleus. So, anteriorly we can see the flagellum, posteriorly here we can see the kinetoplast. So, the undulating membrane these cover all over the body. But in the case of epimestigote form, the kinetoplast is present at the center. It is arise from half of the body. So, it is cover the half of the body. We can see in half of the body. Right. This form is found in peripheral blood of vertebrates. So, example human we can see in the trypamestigote form. Is the diagnostic stage of Trypanosoma species. When we consider the Trypanosoma cruzi, we can say this is the diagnostic stages. This is extracellular parasite. So, it is present in plasma, freely available in plasma. So, by using the flagellum, they can move freely. So, in the case of these four stages, the amestigote form, promestigote form, epimestigote form, and the trypamestigote form, out of these four, amestigote form is intracellular parasite, intracellular form, that other three are the extracellular forms. So, these are present inside the plasma or the liquid medium. Right, this is the morphological form of the parasites. So, the amestigote form you can see here, this is the amestigote form, right. So, other three are, you can clearly differentiate by these three. Here you can see the nucleus actually usually at center, but in the case of promesticode form, it is the kinetoplasm anterior part, flagella also we can see the anterior part. The epistemicode form, you can see the kinetoplasm at center, from that the undulating membrane arises, and again anterior part you can see the flagellum. Again this one, trypanosticode form, you can see the at center nucleus, and anteriorly again here flagella, but the posteriorly you can see the kinetoplasm. Metacyclic trypanosticode form, morphologically similar to the trypanosticode form, but it is shorter and stamped. So when we consider a metacyclic trypanosticode and trypanosticode form, compare with metacyclic trypanosticode forms are elongated, right? It is final development stage in the gut of the intestinal vector and is the infective form from the insect vector to human. So this is metacyclic. Trypanosticode forms are present at the insect vector. This is the final stage, fully developed stage. These are the form transmitted from vector to human. So general life cycle of Leishmania species. So promesticode form is the infectious stage. So promesticode inoculated into the skin when the sandfly take a blood meal. So when the sandfly take blood meal, if the sandfly is infected, it is contained promesticode form. So these promesticode form transfer into the human. These promesticode forms are taken by the macrophage and become the amesticode form. So immediately these promesticode forms are taken by macrophage, and these promesticode forms are converted into amesticode form. Amesticode form multiply by binary fission. So inside the cell macrophage they are multiplying, that means increase the number of amesticode form. These amesticode form are ingested by the insect vector when it take a blood meal. So again, if any sandfly, again same fly or any other sandfly, when they take blood meal, these amesticode form containing macrophage will ingested by the vector. So here again inside the vector these amesticode form become a promesticode form. The promesticode form multiply by binary fission and migrate to the head and mouth part. This is the life cycle of Leishmania species. This is common for all the Leishmania species. Now we will come to the important point, the medical importance of Leishmania species. There are 3 species medically important. One is Leishmania tropica, Leishmania donovani, Leishmania brasiliensis complex. According to the clinical features, we can divide into 3, right? So when we consider these three are causing different diseases. That's why, that means actually the different area of the human. So that's why we clinically we can differentiate these 3 species. So Leishmania tropica we can call all world Leishmania. Again, according to the disease condition, we can call cutaneous Leishmaniasis. 2 type according to the geographical distribution and the reservoir host. One is Leishmania tropica minor, Leishmania tropica major. So Leishmania tropica major is a zoonotic disease. That means this is transmitted from the animals. The habitat, when we consider the habitat of the Leishmania tropica, So we already know there are two stages, amesticode form and the promesticode form. So amesticode form in the endothelial cell of the cutaneous tissue, lymph nodes and the ulcers. So here in human we can see cutaneous tissue and the lymph nodes and the ulcers. So if they ask the specimen, so you have to think about where they present. So tissue biopsy or slit smear, punch biopsy or lymph nodes you can take as a sample. The promesticode form, it is present in the gut of the sandfly. So morphology, it has amesticode and promesticode stage only. So in the case of Leishmania species, all three species, you can see amesticode form and the promesticode forms. So life cycle, definitive host is the male, intermediate host is the female sandfly. Reservoir host, the Leishmania tropica minor, dog, cat, mice. Leishmania tropica major, sand rat example. So the vector inoculate the promesticode into the cutaneous tissue while take the blood meal. So here how it transmit, promesticode transmitted from the sandfly to human. The mode of transmission, so inoculated by the infected sandfly. This is the main mode of the transmission. Second one, direct contact with the ulcers. This is sometime possible to again reinfection, that means auto-infection. There is a possibility and auto-infection. This is sometimes second one, direct contact with the ulcer. Sometimes this is affected the technician, medical laboratory scientist. So when you are handling the sample, you should take care of your work. Then Leishmania tropica minor pathology. This is causes, already we know, cutaneous Leishmaniasis. But this cutaneous Leishmaniasis, when we look at the cutaneous Leishmaniasis, this is dry and normally we can see in urban area, and it is chronic. If it is any lesions are present, it is chronic. So it takes long time to cure, right? And other thing, old world oriental sore we call, right? At the site of the bite there is a dry painless ulcer. So here ulcer in the skin area where the vector is biting. In here we can see the ulcer, but it is dry and painless. Nearly the size is 25 to 70 millimeter in diameter. Usually self-healing after 1 to 2 years. We call already chronic, so it is automatically it will heal after 1 to 2 years. So multiple unhealing lesion known to Leishmaniasis recipients may develop sometimes. So no need touch point. So infection is usually heal and confer long last immunity to reinfection. So here also the automatically it will heal, right? Other one, Leishmania tropica major. This is cause moisture, but the minor is causing dry one. This is wet one, and minor causing urban area. Here this causing rural area. In the case of minor, they produce the chronic one, that means to healing it take more time, but it is acute severe, so it will heal quickly, right, compared with minor. Again this also we call old world cutaneous Leishmaniasis. So again the self healing normally it takes 3 to 6 months. Right, this is enough. Leishmania johnavani, geographical distribution India, Central Asia, China, Sudan, Ethiopia, and you can see in all over the world. But habitat in the case of amesticode form, we can see in reticuloendothelial cell of the visceral organs, such as visceral mean deep organs such as spleen, bone marrow, lymph nodes, liver, kidney, lung, brain, CSF, white blood cells, intestine. So in human, especially in human, amestical form seen in human. So if they ask the Leishmania johnavani specimen, so you have to think the where it is present. Then promesticode form, again, yes, we can see in the vector, phlebotomus, and the leishmania. Right, we all know already the morphology is similar to all species. Life cycle, reservoir host are rodents, dog, fox. So promesticode, again, the life cycle actually is same. Right, but only small difference, the after taking the amesticode form in Leishmania johnavani, it is come to the visceral organs, right? That is only different when we consider the life cycle. Transmission through the bite of insect vector, and again through the blood transfusion, sexual contact, and congenitally, or sometimes organ transplant also here the possible transmission. So here they produce visceral Leishmaniasis. We call other name Kala Asar. So you have to remember this name. So in here, if the liver is affected, sometimes we can see in the hepatomegaly, enlarged liver. If it is affected by spleen, we can see splenomegaly. Right? Remember these things. Right? Prevention and control. Personal protection from the sandfly biting by using nets, avoiding endemic area, especially when sandfly are active. So we have to protect from the biting of sandfly. That is the first things we, then only we can protect. And other thing, if some area is endemic area, especially we have to avoid go that area. Normally we can see in Sri Lanka the Leishmania species and Radhapura, Kurunegala, right? Like the area, the normally we can see. Insecticide spraying of house and farm buildings, so we have to use the spray, insecticide spray to the houses and farm buildings, so then only they can prevent the sandfly, the in-house sandfly. Then destruction of stray dog in area where dogs are reservoir host. We already discussed the dogs are the reservoir host. So normally, so if we identify any of the area in dogs, so we have to destruct the stray dogs. So treating infected person, so here we want to treat the person, they are the source of infection. So if any person identified, we have to treat them. Then we have to give the health education for the community to how it's transmitted, if it is infected, what happened. So we have to give the health education, and we can use the drugs. Right, then laboratory diagnosis, examination of tissue biopsy, aspirate of the spleen, bone marrow, enlarged lymph nodes, and in peripheral blood, especially monocyte. So these are the possible specimen for the identification of the parasite in human. So here you can see the amesticode form intracellularly, or sometimes if the monocytes or macrophage is ruptured, you can see the extracellular form. Again, amesticode form we call LD body or Leishman-Donovan body. So then next method is culture of blood, bone marrow or other tissue where the possible specimen. By using that specimen we can culture, and in the culture we can see the promesticode form. So they may, for example, in MCQs they may ask in culture we can see promesticode form or amesticode form. So you should know in culture what is the stage we can observe or demonstrate. The testings of serum for the Leishmanial antibody. So here you can test IgG antibody or IgM antibody, right, by using known the Leishmania antigen. Formal jet test, it is non-specific screening test for the marked increase IgG. Right. Then and I know I have to talk about the right that we will come to that point. Leishmania brasiliensis complex. Here you can see normally tropical forest of South America, Central America. So here the reservoir host rodents, right? Sometimes some domestic animals. So habitat, when we consider the habitat of Leishmania brasiliensis, here the amesticode form we can see in human in the reticuloendothelial cell of the mucocutaneous tissue of the nose, mouth, lips and larynx. So these are the area in human we can see the mucous cells. So mucous area. So here in the case of the Leishmania brasiliensis, they favor to live or habit in mucocutaneous area. In human normally where it is present nose, mouth and lip and larynx. The promesticode form normally it is present in the vector. Here in the case of Leishmania brasiliensis, the vector is the Leishmania sandfly. The morphology again same like other two Leishmania donovani and the Leishmania tropica. So when we consider the life cycle, here the vector is the Leishmania sandfly. These are the main vector. So again life cycle is similar. Here pathology mucocutaneous leishmaniasis, chronic ulceration of mucous membrane of the mouth, nose, throat. So here we can see the chronic ulcers in the mucous area with destruction of bone, and sometimes it will destruct the bone and cartilage. So prevention, again avoid the endemic area, avoid the insect bite and treat the infected person. Health education, right. Laboratory diagnosis, here also we can identify the amesticode form in the specimens, especially ulcers, lesion or sore or nodules, by using stain like Ginsar stain. So in culture we can see the promesticode form, immunological studies and the Leishman skin test. So here the common thing, the clinical diseases with visceral leishmaniasis caused by Leishmania donovani, cutaneous leishmaniasis caused by Leishmania tropica. So in the case of Leishmania tropica, again we can divide into two, Leishmania minor, Leishmania major. In Leishmania minor, it produces dry urban cutaneous leishmaniasis which are chronic. It takes 1 to 2 years. But major, it is wet rural cutaneous leishmaniasis, but it is acute. That means 3 to 6 months it takes to heal. Mucocutaneous leishmaniasis, again we can call New World mucocutaneous leishmaniasis. or sometimes we can call American cutaneous Leishmaniasis. So these are the things here we are discussing. Another thing I need to talk about in Sri Lankan situation in Leishmania species. So in here Leishmania species in Sri Lanka, normally there are some endemic area like Anuradhapura. So in here we can see the normally in Sri Lanka there are so many cases reported as a cutaneous leishmaniasis. So it is the major leishmaniasis, but it is caused by Leishmania donovani. It is caused by Leishmania donovani. But other countries, Leishmania donovani produce visceral leishmaniasis. But in the case of Sri Lanka, Leishmania donovani, Leishmania tropica both produce cutaneous leishmaniasis, and the vector is Phlebotomus argenteipus, right? And laboratory diagnosis and other things here in Sri Lanka, you have to consider as the cutaneous diagnosis, not a visceral one. If it is visceral leishmaniasis, you have to think about the deep organ, liver, spleen and other things. But this is cutaneous, so skin sample, split biopsy, slit biopsy, punch biopsy, and the split smear, impression smear. These are the possible smears you can take. Right.